To the University of Washington Data Science Seminar. As part of the UW, we would like to acknowledge the Coast Salish people of this land, peoples of this land, the land which touches the shared waters of all tribes and bands within the Squamish, Tulalip, and Muckleshoot nations. The first 4 of these data science seminars, so this starting this week and the next 3 weeks after this, are going to be from participants of the eScience Incubator, which is a program that we run each winter quarter, where we pair researchers from around the university. We've had projects from more than 28 departments across the university over the 7 years that we've been running the program, with data scientists and research scientists from eScience to do a kind of a deep dive on a project where we feel like we can really make progress only by in that collaboration. So it's really a collaborative program where the data scientist goes kind of deep into the project. It's a 10-week program, and the data scientists and the research scientists and the project leads spend 2 full days a week for 10 weeks working together closely. So if you are not aware of the incubator, you would like to know more about it, please look it up on our website. We have sort of little blurbs from each of the previous projects we've done over the last 7 years. And keep your eye out next winter if you'd like to join. We have some info sessions beforehand, and we'd be happy to talk to you guys in office hours. Are a really great place to come and talk and kind of scope a project to see if it would work well for an incubator. So I think that's all the intro I'm going to do in the interest of leaving time for the talks. So Sarah's going to introduce the speakers.
Great, thanks, Brenna. So yeah, we're delighted to have 2 of our incubatees presenting today. And the way this will work is so we'll have 2 short talks, and each short talk will be followed by several minutes of questions. Because we have those questions interspersed, please drop your questions into the Q&A throughout the talk, and then we can address those when each of the speakers finishes. So we're going to be starting today with a talk by Dhruv Balwada. He is a research associate in the School of Oceanography, and he collaborated with Rob Fotland and Scott Henderson with the eScience Institute. And Dhruv, I'll turn it over to you. You're muted.
What about now? Yeah.
Sounds good. All
okay.
set.
Yeah. Thanks, Sarah. We'll start again. So I'm in the School of Oceanography, and over the last 2.5 months, I worked with Rob and Scott from the eScience Institute on mapping and visualizing ocean gliders to observe submesoscale flows. And if those words don't make sense, I'll try to explain them to you soon. So the structure of the talk, I'll first introduce the problem, why we study the oceans and the submesoscales and how we go about observing them. And then I'll go over the 2 ideas that were pursued as part of the eScience Incubator. And finally, I'll talk a little bit about ongoing and future work. So there's many reasons to study the ocean. If you care about fish, maybe that's why you want to study it. Or if you care about coastal climate, you might care about the ocean. But two of the reasons that I'll be talking about are to do with climate change, which we know is happening, and humans have been causing it for the last 150 years at least, very drastically. And so during the last 150 years, what the humans have done is that they have put in a lot of carbon dioxide into the atmosphere. And what happens to that carbon dioxide matters for what's going to happen to the climate. So of all the carbon dioxide that we have put in the atmosphere, about 30% of it has ended up in the oceans. And that's shown in this plot over here. So basically the curves going up are the ones showing you how much carbon dioxide we have added in. And so there's a lot. And the bottom curves show you that, the dark blue is that there's about 30 or 25 to 30% that's gone into the ocean, and about 30% in the atmosphere, and about 30% being stored on land. So what this means is that essentially the oceans have taken away about 30% of the carbon dioxide that we have added in into the system. But there is still 30% left in the atmosphere. And what that does is that it causes the Earth to store extra energy that it wouldn't have in the past. And that energy itself goes into the different components of the Earth system. And so about 90% of the excess heat or excess energy has ended up actually in the oceans. And only about 10% is kind of split between the land and atmosphere and ice parts. So there's all this extra heat and extra carbon in the Earth system, and the oceans kind of played an important role in buffering or mitigating the role of humans. The plots over here show you where all of this extra carbon dioxide and extra heat has gone. And basically you see that it's not being taken up by the ocean uniformly. There are different regions. So there's parts of the North Atlantic over here, which have taken up a lot of carbon. And there's a huge band over here in the south, which we call the Southern Ocean, which has taken up a lot of carbon. And in a similar fashion, the North Atlantic's also taken up a lot of heat, and so has the Southern Ocean. So why did the Southern Ocean take up so much heat? And this can be explained by the special structure and the circulation of the Southern Ocean. So one thing that I just want to point out is that the Southern Ocean is the only place in the world where you can go all the way from east to west and not hit any land boundaries because of the opening between South America and Antarctica. And what that does is that it sets up a pretty unique circulation pattern. So just kind of focus on this panel over here that's towards the observer. And this is showing you a section going from south near Antarctica towards South America. And up and down is depth. And basically what you see is that there's water that's kind of moving towards the south, coming up towards the surface. And then some of it sinks back in the northern side, and some of them it sinks back on the southern side. But basically what happens is that you bring in this water from depth, you make it interact with the atmosphere. And when that interaction happens, the water exchanges both carbon and heat with the atmosphere. And then it goes back down to store it. And another important property of the ocean is that as this process is happening, a lot of this transport takes place along sort of constant temperature or constant density surfaces, which are shown by these colors over here. So when you're in the north, the temperature changes quite rapidly with depth, and that doesn't allow heat or carbon to move very far into the ocean. But when you're in the south over here, you can exchange heat and carbon, and then that can slide down along these constant temperature surfaces into the deep ocean. And so that's a little bit of a cartoon of what happens in the Southern Ocean. This is results from a simulation that we ran. And so on the left-hand side, again, you have a cartoon version of the Southern Ocean. The colors don't matter, but the point is that these density or temperature surfaces rise to the surface as you go to the south from some depth. And what I'm showing here on the right-hand side is basically a movie of a tracer, which could be carbon or heat getting mixed into the ocean along a pillar surface. So the density surface I'm showing corresponds to 3 degrees Celsius, and that's this density surface that rises up to the surface, of the ocean. And what you see is that as time, so initially there is no, when we start out, there is basically no tracer at depth or in the interior, which in the interior over here means that in the deeper parts of the ocean. And then we start to put carbon in, and as time goes on, all this complex circulation mixes up that carbon into the deeper parts of the ocean. Now, this result is from a simulation, so we want to see if any of this is actually happening in the real ocean. And to do that, we have to go and make observations. Also, you can see that it's a pretty complex process. It's got a lot of scales. There's a lot of motion going on. So how do we observe this? And so to observe it, there are many ways, but the one that I use are these instruments called sea gliders or gliders, which are basically ocean robots that go up and down, and as they move through the water column, so this is the surface, and they go down and then they'll go up. and as they move up and down, they will collect measurements of different properties. So this can be temperature or salinity or how much carbon there is in the ocean. And every time they come up to the surface, they will transmit their data back to the satellite. And then as the controller, we can also tell them where to go next. So that's what the sea gliders do. So the sea gliders that I was looking at, that I've been looking at over the last year or so, were released in the Southern Ocean. So that's Africa over there, that's South America, and that's Antarctica. So there's this little patch of land over here where we put our sea gliders in. And this is a blow up of that same region. And what I want to point out is that over the 3-month deployment period, the glider sampled a region which was about 500 by 500 kilometers, and it had a complex sampling pattern. So, you know, it kind of went straight, did some loops, went straight. And part of the reason that was happening was that we were asking it to go somewhere, but then sometimes it would get stuck in really strong ocean currents and we couldn't control it. Then it would come out of them and then we could make it go straight again. And so there's a complex sampling pattern that's going on. And so let's see at what data we get. So on this top panel over here, I'm showing you a part of the temperature measurement that was made by the glider. And so the glider kind of starts out and it does its up and down zigzags as it goes along from the surface all the way to 1,000 meters, up, down. And then this plot is made as a function of distance that the glider travels. So there is this initial part when the glider is kind of going straight, and then it gets stuck in a current and it ends up slowing down quite drastically. And so what happens during that slowdown is that the distance between the up and down profiles decreases. So you're sampling smaller scales. And then at some later stage, the sampling profiles move apart again. And so you start sampling larger scales again. And what I want you to take away from here is that the data has a very complex sampling pattern, both in terms of this depth, pressure up and down, and also in terms of the trajectory being over, you know, sort of a complex trajectory that's taken. And the second part is that there is variability present in the observations, for example, of temperature that are being made over a wide range of scales. So, you know, there are these big blobs, then you look in, there's a lot of small features that we are interested in. And so this kind of gives us two tasks at hand. One is that we want to come up with a mapping or an interpolation algorithm that will help fill in gaps, or in other ways, take this data from this very non-uniform scattered grid to a more gridded product, so that we can do things like plotting it more easily, or maybe if you want to compute the gradients of some quantities, we can do that. And the other bit that we care about is to be able to interact with this data and visualize it, so that, you know, if I wanted to zoom into this feature to see what was going on, I can zoom in, change the color scales, and all those things on the fly. So those were the two tasks. So first I'll talk about the mapping problem. So traditionally what's been done in oceanography is that you either do a very simple interpolation, which might be like linear interpolation or cubic splines. And people who want to be a little bit more fancy might do something that's called kriging, which I'll explain now. But in that, you have to choose. Okay, so let me just explain what. So if you want to be a little bit fancy, what's done is kriging. So this is sort of a flow diagram which shows you what kriging or Gaussian process regression is. So let me first explain what kriging is. So in kriging, you take some scattered data. You assume that there is some correlation structure, which means that points that are close together are related to each other in a certain way. And the traditional choice that's made is an exponential function, which basically says that if the points are far apart, they don't know anything about each other. If they're close together, they know quite a bit. And then there's a length scale that's chosen to go with the correlation function. And that's called kriging, where you choose the correlation structure and you choose the length scale. And then you get a mapped data with some uncertainty estimates. What's done in Gaussian process regression instead is that, first of all, you don't choose the parameter. You fit it probabilistically using a maximum likelihood estimation. And you can also play around to not have to choose the exact correlation structure. You can sample through a variety of choices. So just as you would choose a length scale or a parameter, even your correlation structure would be chosen in a maximum likelihood kind of fashion. So why does this correlation structure matter? Right. So over here on the right-hand side, I'm showing you kind of two examples. So the top panel is interpolation that's done using smooth correlation functions. And the bottom panel is the same thing done using a correlation function that tells you that the functions that are being fit are rougher. And the little red dots are the places where we have samples. And so basically you're trying to kind of fill the regions in the middle. And what this tells you is that there's a bunch of functions that can go through the observation points. And depending on the correlation function, these fitted functions can either be smooth or they can be rough. And all of this matters for things like uncertainty estimates, or if you want to compute gradients. Now, why do we want to move away from the Gaussian kernel function, which would give you something like this? Like, why do we want to go to something rougher? And that can be seen in this picture over here. So if the power spectrum is one way to quantify how the structure of the function looks. And just focus on the black line and the blue one for now. And so the blue line is the power spectrum of the observations from the glider. And so what it shows you is that there is some sort of scaling that's going on, and it has a certain slope. If I was to choose a Gaussian kernel, what that would do is that it would force my interpolation to make something that looks really smooth. And so basically it would end up smoothing out a lot of the variance that's there in the data. And so I hope that that gives you a little bit of a summary of why we want to do Gaussian processes, but I just want to share the results that we got. And this is still preliminary work. We're working on it. And so on the top is a part of a glider section. So you can see these little up and downs. Over here is what you get from linear interpolation. So when you get a map, it looks okay. It can be very rough. And when I start to compute gradients, I will run into problems. Now, if I was to do Gaussian process regression, that's shown in the bottom panel over here. And there are 3 panels, and the 3 correspond to 3 different choices of correlation functions. One is the Gaussian or the squared exponential. There's something called Matern 3.2 and Matern 1.2. And the thing that's shown in the parentheses are the maximum likelihood estimates that go with them. And a higher value means that you have chosen a better model. What I'm showing is that if you take the standard way versus if you choose these Matern correlation functions, which approximate your data or model your data better, you improve your maximum likelihood estimate. So this is a better model or a better fit to the data, and hopefully also a better prediction of what's going on in the intermediate points. So that's all I want to say about what we did with the mapping. I just want to give a shout out to Gaussian Processes Summer School, which has this website called gpss.cc. And they have a package called GPy, which is what I used to do this. And if you want to learn more about Gaussian processes, definitely go check out this website, because they have really nice tutorials collected over the years. And these are really the experts in the field trying to teach you in very simple ways how these processes and this machine learning technique works. So the next bit has to do with visualization. So there's 2 traditional approaches. This is not specific to oceanography. This is basically if anybody who's interacted with data. There's the first one, which is what I call the code plot approach, where you write some code, you make a plot, you don't like it, you write more code, you add something, you write more code, you change it a little bit, then you realize you don't like something, you go back and you keep going back and forth. The other approach is where somebody made an interactive graphic user interface for you. And in that case, it's pretty nice. You can play around with widgets. It does a lot of things. But the moment you go to one of these things, usually you wish for that one feature that it doesn't have, right? So what we wanted to do was take something that's in the middle, which is the code interactive plot algorithm, which is, and so that's what we did. And we used this set of libraries in Python called, that live under the umbrella of something called HoloViz. And basically HoloViz is an ecosystem of these 7 libraries called panel and so on. and each library does something, but all the libraries can talk to each other, which allows you to make interactive visualizations with relatively little Python knowledge. And so you can make an interactive visualization pretty quickly, change the code if you don't like something about it. This is a slide from their website showing you the Holoviz philosophy, right? So they say that there's 3 ways to do it, and the layered approach is what Holoviz does. But the first one is the completely low level one, where you write some code. So the point is that you're trying to go from this point, the little triangle, to the little circle. And in the standard code plot approach, you would write some code, make a plot, and you kind of just meander your way through. The GUI approach would be something high level, where somebody has made something for you, and maybe it takes you where you want to go, but you don't exactly land at the point that you want to land at, right? You might be a little bit further away from it. Like your GUI is not doing exactly what you want. And then there's the layered approach, where you write like one function, it makes a GUI for you with your data set. It's not exactly what you want, but then you can start to dig into these 7 libraries and get to the exact point that you want. So now I'll show you a little demo of the visualization platform that we created, which is over here on the right. There's this little barcode that you can scan if you want to go to the GitHub repo that has all the code in Jupyter Notebooks. And it also has binder links that you can just click and the dashboard would open up and you can interact with it. And even though the final product right now is a dashboard, what I'm showing you, that was not the purpose. The purpose was to provide an example of code that people can dig into relatively easily and adopt this dashboard to their own glider data, or maybe they have some other observational oceanography data that they can relatively easily play around with. So here is the dashboard. Right now it's running on my own computer. There's basically 3 parts to it. There is there's the place with the widgets, where you can choose things like different variables. And there are 2 main plots. There is the first plot, which is lat versus lon, and it's showing some field in the background, and it is showing the trajectory of the gliders. And it's also overlaying on a map, so you can zoom out and see where exactly your experiment was. The field that's in the background is sea surface height, which is the field that we care about, but you can change it. You can change it to, it's anomaly field or something. And we had 2 gliders, so you can switch back and forth between those gliders. You can choose the range over which you want to make a plot, and there's a little time slider over here that allows you to do it. So right now it's plotting from day 120 to day 164. So you basically plot more of the trajectory. And the section of the glider that goes with the trajectory is being plotted here in the bottom. And you can choose the different variables that are measured. So right now it's plotting temperature, but you could plot temperature, salinity, density, oxygen, chlorophyll, and whatever you wish. Then the other thing is that so I plotted this. There is a lot of structure here, but it's all white, kind of looks white, right? Because I don't have the right color scale. But I can very easily go in and change my color scale to look at this feature, and then maybe I don't like it, so I can refresh it a little bit. The color scale will readjust. Yeah, there it goes. And so there's a lot of things that you can do with it. I can't go over all of them right now, but there's a lot of options. But these are the options that I chose to build in, because this is the kind of analysis that I wanted to do with these gliders. And kind of just tying all of this back to the things that I was mentioning about submesoscales and how tracers get mixed. Well, what we're starting to see already over here is that this is temperature, which is one tracer over here, and there are parts which are sort of cold, warmer, and there are parts which are colder. And some initial analysis has kind of revealed that these little blobs over here might actually be associated with tracer moving from the surface of the ocean into the interior. And now I can study that using this visualization platform, kind of look at exactly where this came from and where it sits in my data set. So going back to my slides, this is just, so okay, so.
just letting you know, we need to wrap up. We're about at time.
Okay, cool. Yeah, so I just have one. This is the last slide.
Oh, okay, great.
Yeah, just wanted to give you a little bit of the basic structure of how the dashboard is made. So it has three parts. There's the widget parameters part, there's the trajectory plot and the section plot, and they all talk to each other, and that's built in. And then there's a separate section where you have to clean your data and format your data and interpolate your data. And all that gets fed into this as netcdf files. And there's obviously the part that came before it, which was when the glider was deployed, the data was collected, quality controlled, which was fed as netcdf files. And some of the things that we're looking into right now is trying to maybe use this visualization platform to actually move the glider around in real time. So if you want to know more, you can go to the repo, and just have some future work listed over here, but it's not important to go into it. The only thing that I want to mention is that because everything lives on GitHub now, people are already starting to interact. We have a wish list, and they're opening issues. And so hopefully this dashboard kind of grows from here. Yeah, so thanks. And if anyone has any questions, I can.
Great. Yeah, thank you, Dhruv. We have a question that just came in. I think we have time for probably about one question. And the question is, well, it's a comment first. That dashboard is sweet. It looks like your plots are interpolated on a plane, 2D, with time on your x-axis. If your recording robot was moving up and down and side to side or backwards, and not just in a straight line, will you soon be able to interpolate in 3D space?
Yeah, so I think the person, they actually said that it's on my wish list. So yeah, it's on my wish list, and that's actually the next thing that I want to do is, so yeah, the HoloViz world allows you to make interactive plots that are also 3D, and so you can connect them, and so that's something that you care about. Yeah.
Great, and you can probably see we can take one more question. We have one. Nice presentation. How intensive is the data cleaning and quality control from the gliders?
It did take a lot of time. So that is not the bit that I was doing as part of the eScience Incubator. That's what we did last year, and there were multiple of us who were kind of working on. But there's another library that we have, which is called Glider Tools, which has been built to do that. So if you're interested, you can email me or try to search on GitHub for Glider Tools. You'll find it.
Okay, and I'll just actually want to take a quick one here. How did you verify that the selection interpolation method was effective?
Yeah, so that's a good question. So the way that it was done in the case that I showed you was we just used the maximum likelihood estimate as a measure of how good the interpolation was. But if you know more about this, I would agree that that's not necessarily the best way to do that. And so that's also in future work, that, in what sense did we do better than linear or cubic interpolation? And that's something that I'm working on right now. I just didn't get to it in 2.5 months.
Yeah. Okay, Dhruv, I'm going to ask you, and you can probably see the last question there, which you could answer, but maybe while we're transitioning, you can answer that, and yeah, put down your slides. And Aji, if you want to go ahead and bring your slides up.
Thanks, everyone.
And yeah, I'll just let you know. So our next speaker, thank you so much, Dhruv. And yeah, if you want to answer, I know there's a couple additional questions, and maybe Dhruv can use the Q&A functionality to answer those in text. But next up, our next speaker is Aji John, and he's a graduate student in the Department of Biology. And he worked in the incubator with Nicoletta Christie and Amanda Tan. And Aji, I'll turn it over to you.
Yeah, thank you. Hope you can hear me. So from oceans, we will come to alpine wildflowers. So very much this work is in progress. I have put my email in there. So if I'm not able to answer your question, feel free to send me an email. Yeah, we'll start now. So I wanted to thank numerous people, including the eScience. I've worked with Dr. Nicoletta Christie and Dr. Amanda Tan. and there've been other collaborators as well, but it was truly a good partnership to get this rolling. Again, some of the things which I have explored is that support from AI for Earth. You should apply if you get a chance. It's from Microsoft. So just to set some sort of context, I want to show you this meadow, which is at Mount Rainier. And this is our study system and why we are interested in it. So you will see, I'm showing you kind of the topography as well as the vegetation. It's a video was taken by somebody in YouTube, and which we were able to use that as a context that, there are these flowers, and flowers especially give you a sort of a good context, and because they're good context in the sense of climate change and the impacts to climate change. Because these alpine species are very sensitive to climate, we will quickly see how, or we can gauge quite effectively what's the impact. With this, are they shifting the flowering? Are they not flowering? And you can see how diverse these meadows are. Kind of we have around 17 or 18 species there, and you can see how these rich meadows are actually quite a good study system to actually work with. So, and typically when we look at these meadows, we are interested in phenology, which are the reproductive stages. And we look at budding, flowering, fruiting. These are different stages, and each one is important. Flowering especially, is of interest for us because it's a very dominant signal. And obviously, as a visitor, if you have ever been to Rainier, it's quite a nice view as well. So a lot of this study focuses on flowering, and flowering to us, in our lab, which is Janica Hilbers-Lambert's lab in the biology department, we have been studying for the last 8 to 10 years, and it has given us numerous insights into into how climate change is affecting these alpine systems. And so just to set kind of the domain here of the study system and what my research interests are. So I am interested in pretty much, what happens to these alpine meadows because of the environment. And here specifically, specifically snowmelt. And the way we, and to actually set further some idea into what these meadows are and how these are how we typically visualize them, is that they would have a peak display, like a peak floral display when most of the species in these meadows flower. So and that is quite, it can be detected using, like manual observation. We have seen it to be observed by satellites as well, with different resolutions. But to study them, the drivers of these meadows, first of all, the flowering and the composition of the species are complex. They depend on snowmelt. I mean, when the snowmelt happens, because that's when the ice melts, so the snow melts, and then these perennial species come out again. They vary by elevation, which means, at a lower elevation, you would find less number of species compared to the sort of mid elevation. And here, sort of elevation spread is around 1,400 meters to around 900 meters, where you can find these meadows. And the last one is the temperature. So if it's a warmer year, you will see these shifts happen. Either they will sort of shift their flowering earlier on, or they will delay it. And where we see them is that when this multitude of these species flower, we get the unique sort of flowering phenology curve. What we say is that this sort of the culmination of these flowering, and they happen to last for rather 2 weeks or so, and that helps us to sort of put a yardstick on, okay, this was the flowering on this year. And typically, not typically, I should say. It is quite important because to study these, because there are downstream systems like pollinators or other insects which use the same meadows. And so any sort of disruption in this flowering phenology would have, might have catastrophic impacts. And the way we have studied in our lab is normally by citizen scientists, where folks go every year, even they're going to go this year as well, or by researchers who go focus studies at these meadows. But the challenge with all of this is that it's a very tough, sort of time-consuming effort to do it, to actually observe, to tally if they're flowering, not flowering. And we tend to miss a lot too, because we tend to go on these trails and then they only can go, they can't go into the meadows. They can only see from these meadows, from these trails, what's flowering and then make a note. So there's chance that we can miss it. If it's inclement weather, we can't go. So there's tendency to miss it. So last year we started a project with the understanding of, can we use sort of fine scale remote sensing? And I'm including rather cameras and drone imagery, not drones, especially you can't fly drones in the national parks. But the idea would be that at some point, can we actually pick these flowering patterns? And then focusing only on the, when I say drone, think of it as a drone camera. And then pick up these flowering signals, like peak flowering signals. And the way we did it was we went there last year and we took a drone camera and a phone camera, and we took pictures of these meadows, around 6 to 7 times through the flowering season. And we hoisted like a drone camera, which could give us spectral information. This project has not used the drone imagery yet, but it has used the other similar imagery for this project. And then the idea is at some point we'll connect with other satellite imagery so that whatever we are finding in terms of the flowering signal, does it correlate with a satellite imagery, which is planet, which is a 3 meter, or with the other coarser one, which is like a 10-meter resolution. And the intuition behind is that why we want to link both of them is that, if there are spectral signatures, they are driven by the flowering colors. So, and the idea is that if we have these when we have these satellite images, we can pick up the same spectral reflectances, which would say that which flower, is this meadow flowering, was not flowering. So this is showing you like a sample meadow where the number of species which we have are like, I've shown you only 7, but there could be more. And but before we got to that, to actually compare, we wanted a way to actually segment or detect these flowers so that we could start counting them and say, okay, this particular meadow is in peak flowering, and these are the species which we see, so that we can validate. So we can validate that, yes, in fact, it is flowering and it is true, and we can validate we can validate so the richness, which is the unique number of species which you see in each of these meadows, because because they do vary by elevation. So this at the eScience we explored these convolutional neural network based approaches to detect these flowers, which could be very small in scale, and find out a way to train and do the evaluation. So just to reiterate the point, what we wanted was to go, instead of showing you a single flower by saying this was mountain daisy, we wanted to look at a picture by saying and find out all the mountain daisies, all the magenta paintbrushes, all the Sitka valerians. And to actually do that, we did some research, and we found out that the 2 methods which folks have been using, which is Max RCNN and Yolo, these are all used very much in object detection, and they are pretty popular even today, and they've been improving quite a lot. Yolo is from here, from my home university, University of Washington, and I'll briefly touch on what these methods are. So Yolo works. Yolo is a single shot, which means that it takes an image, it runs a convolutional neural network on top of it, and then it actually produces the predictions for the individual objects it has detected. It is fast. It is normally, it's considered to be very generalizable, which means, let's say, if you're running it on flowers, you're going to get a very good kind of transferability in, let's say, doing cell detection. It has been used. There's a lot of versions of it. It's not, but it has, and also it has, it is quite good in terms of performance. And the other ones which we explored was a mass-CRNN, which is now, part of the Facebook the research program, but which is a two-stage network. So there's a head part and the tail part, if you want to think about it. And the head part is a convolutional neural network, which you can swap out, like if it's, and you will see. I'm not going into details here, but actually feel free to check the web. You can change the head, which means you can change. Like the ones which we have played is called ResNet, which is a convolutional neural network, which could be very dense, and that can be used for like feature detection or feature learning. And then the second part of it is the detector, which basically fine tunes the detection. And in mass RCNN, it also gives you the masks, which means the pixel level, at pixel level, which one is which. We have not used the mask RCNN part. So these are the two methods we explored. And I want to show you next is how the detection worked with one algorithm. So this is with Yolo. And I'm going to show you, you can see, so right up front, we had a problem. So you had seen that it's classifying a rock as a Sitka valerian as a flower. But it's doing very well in other areas where it's able to detect pretty good accuracy in terms of the uniqueness of species and the number of species, but it fails in picking up all the instances. Like there's a good example where it's doing much of it, like in focus, it's doing quite well, but the ones far away, it's not able to do. And so this, what we did was we took a video and we ran the algorithm on each and every frame, and then we stitched them together. So yeah, I mean, this way, there's a lot of room for improvement. We have not done a similar kind of video with mask RCNN, but next I will show you some of the slides which go into the focused kind of issues or the good things which I was able to pick up. So this is one where I'm showing you with the mask RCNN, where on the left-hand side, it's the original one, and the right-hand side is the predicted one. So here it's able to pick even more than what we have labeled, so which is great. This is where it's, like the similar one is lastly one, it's able to pick more than, which is good. It's always, if a model is able to detect what we missed in the labeling, it's in our annotation is good. This is where it's similar problems with like where we're not able to pick up all the instances. So this is a problem which we are trying to work through, but where you can see that, there's a rather subalpine loop and it's able to pick some, not the others you can't. I'm showing you like a big frame detection. Yolo is able to do it better, where you can feed in any kind of image, any resolution image, and it's able to do pretty well. But similar with Yolo, if there's very small, then it can't detect. So this is a horizon problem where the ones in far away, it can't do it. So again, with Yolo, it's problem with rock is Western anemone, which is one flower which we have to work on. But it also does pretty good with the ones in the shade. It's doing well, but if it's doing well in shade, but it's not really doing well in the other part of shade. So here you can see Cascade Aster, which is doing quite well in some parts of the image, some are not. So I'm just going to quickly compare between Yolo and mask RCNN. Again, just keep in mind that this is work in progress. We have not explored all the tunings possible. We're still learning quite a lot. So we are just comparing with Yolo and mask RCNN. So where we find generally that mask RCNN is able to pick up more instances. So even smaller instances it's able to pick up. And more instances and more kind of resolutions as well. When I say more resolutions, I mean, say, even if I feed in a smaller tile size, let's say a 256 instead of, let's say, 25 by 25 or 100 by 100, it's still able to do better than even rather compared to Yolo. So then next we said is like how does it work with Flickr data sets, which means Flickr is an online sharing site where you can post pictures. And we ran the whole, just YOLO on the Flickr. And then this is how it worked, where it was able to do quite well. There is a constant, it's not a nagging problem, but it is a problem where there's obfuscation. Like here, there is one flower on top of the other, and there's a butterfly sitting on one of the American bistrots. It's not able to detect, but it's able to detect the one which is blurred, which is very good. Able to do quite well here, even with the background of an animal, it's able to do well. And when it's in like a blurred, it's able to do well. And you will see why it was good in a way that it didn't have any training with kind of blurred images, but it was still able to pick it up. It does quite well in the Flickr data set, things which are much in focus. Like if there's only one image with just the picture of the flower itself, it's doing quite well. But we see the same problem in the Flickr data set where, it's picking up, again, this is not an easy problem, I would say that, like Grace Lavage comes in these clusters, but like Western anemone, this is not really a flowering Western anemone, but this is what we use for training, but it's able to, at least detect one. Constant issues where, again, rather, this also gives us, using Flickr dataset is actually good now because now we can use our training to actually improve our training, because we have a lot of class imbalance issues where, we had more of, where we had relatively few of cascade aster, what you're seeing is avalanche lily on the left two images. But these are the problems which we've seen otherwise in the other in our own test set as well. So from the ecology point of view, we ran it on 1 month, which is August of last year, and it's able to show that kind of spread, where we would see that there's more diversity on the middle elevation, 700 meters, and there's less diversity on the extreme sites, which are 400 meters to 900 meters. So this is a good, rather support for the hypothesis that yes, it's able to pick it up. Now the thing is that we would be trying it on other months to see if this holds up. So just to recap, what we found out was that to these kind of heterogeneous meadows, we were able to try two convolutional neural networks, which showed pretty much a good degree, of the classification or detection accuracy, and we were able to make a first pass at getting sort of the phenological peak, if you want to say, or a flowering peak. Some of the key things, rather takeaways, is that it's a good learning curve, and what we found out that masked RNN seems to be better, and there was a lot of caution in the beginning saying that detecting small flowers would be difficult, but we managed to do it. So what we found was the key was the tiling. So instead of doing a big, 1024 by 1024, you just go a smaller tiling. We saw that the image augmentation, which is when you take the training image and you skew them, you do various the image enhancements to it. That is differential. We saw that masked RNN seemed to be benefiting more, YOLO not. Something which if you folks are interested, what I've seen is that whatever we have done is off the shelf, and I think there's something from Facebook which is called Detectron 2, which think of it as a prefab for your machine learning. You can plug and play, use that, that's great. You don't have to reinvent the whole thing. You don't even need to understand what a convolutional network is. Colab soon, don't stick too much to Colab. It has a lot of computational restrictions. When you start scaling up, you will see that there's a huge bottleneck to your whole development. Azure Machine Learning is good. You can get things through our university. To scale up, use online GPU offerings. What I mean by the companies like Lambda Labs, which gives you, like a rather $1 an hour for like 10 GPUs. Use that if you want to do quickly check your model. Some tuning, I said, as the next steps. I've been very vague here because there are a lot of tunings you can do, things which I do understand, things I don't understand, and to do actual correlation of satellite imagery. And now next thing would be to release to the citizen science folks. That's about it. Feel free to ask questions in the chat.
Thank you, Aji, for your presentation.
Sure.
Um,
Okay, I have.
there are two questions here. One is from Carly.
Mm-hmm.
Hi, Aji. It is so cool that image you're IDing can be applied to white flowers. What did you use to train the CNNs? Images of the flowers from all angles? What wavelengths does time of day of the pictures affect correct classification?
Yeah, excellent question. It does affect the time of the day. They were all taking around noontime, and all the angles were taken sort of from the waist height, or some are waist height, some are at the sort of shoulder level, and they were all looking down. So that's reason, what we've seen is that a lot of things which are worked in R is that people, when they're posted on the Flickr, they're all taking these pictures. However, the drone imagery were taken from around 11 feet or so, looking down on the meadow, at the same, like a skewed angle. And we used, like camera imagery to actually train this model first. And this was just your iPhone image to begin with. And then we tiled them. We tiled by 256 by 256. First, we trained the entire image. That didn't go very well. Then we tiled them into 256, and then we trained it that way. So we annotated each tile by saying, okay, this is a magenta paintbrush. These are instances. So we put bounding box in each one of them. Similarly, we did for all of them, and then we trained it that way.
Thank you, Aji. Here's another question from Rebecca. Mm-hmm. Really neat project. Those Western anemones are such unique and lovable flowers. I think we all agree with that. Yeah. Question, at this point, if the detection identification is errant the same way for all images through time, might you still be able to tease out ecological changes over time going back through historical photos?
Yeah, sorry, can we do the last part once again? I didn't catch the last part. Sorry, I should.
Okay, so we're
yeah, sorry,
at
I
this
can't
point, yeah, if the detection or identification is errant the same way for all images through time, might you still be able to tease out ecological changes over time going back through historical photos?
Yeah, great point. What we are primarily interested, there are two folds to it. If we can detect the unique number of species, I'm not that concerned about rather sort of missing on the unique instances. So unique species. If you're able to get that, I think we can still come to the same conclusion. Actually, the number of instances of a particular species only confirms that point. So we are actively working on trying to fine tune it, and we're getting confidence more in our kind of tuning. So we haven't shown you, we were able to get some more tuning, some more performance boost on it. I haven't shown it, but yeah, but we are confident so far. So we are trying to get last 6 years of Flickr data and see the same thing. So we have just sampled only 400 so far, but we have around 4,000 to go through. So.
Looking forward to that. Sarah has a question too.
Yeah,
Sarah.
please.
Yeah, thanks for a great talk. I was just wondering, there's a lot of little small white flowers. I'm just wondering, how does this bias towards sort of large charismatic flowers, and how well do you capture kind of the small flowers that maybe even like there's some flowers I know that are so small that they hardly even look like flowers. So how well do you do on those kinds of species?
Yeah, that's a good one. I mean, American Bistard is the one which you saw, and when they are not in focus, it's not able to detect at all. It's very crowded, and they tend to co-occur a lot, and with other ones. So yeah, it is a problem which we're trying to work through. And so there are a couple of ideas which we're trying to actually blow up that image and try to pick the patterns. And because even if you look at it, I think the one in Horizon, I'm not that hopeful, but at least the one closer, we should be able to pick, like the intricate patterns of the flower. But it might come down to actually we annotating better. Like what we have shown is that we are not good annotators, so which means that we have to do a better job at trying to annotate what a Western anemone is, what an American bistort is. The one thing which we have done in our first step here is that only bounding box, which you just draw a boundary. The better one way is to do the what they call is actual masking. So you do a polygon and you kind of curve, and that's the better way of doing it. It's just a lot more time consuming, you need a lot more people, and that might actually help do this better. So yeah, great question.
Well, thank you very much, Aji and Dhruv. Those were excellent presentations to start our seminar with. And I think we can conclude our seminar for today. And we'll see you next week.
Okay, thank you so much.
Thank you.
Thank you.